One of the factors that will greatly speed up gene therapy is faster and cheaper genome sequencing as computing power increases. Prices are falling rapidly. The first human genome project cost $2.7 billion. Until recently, it cost about $2 million to sequence an individual's entire genome. But a new biotech company is promising to be able to sequence your individual human genome for under $5,000. In a few years' time, it's predicted that sequencing a person's entire genome will cost just a few dollars. At that point, everyone will be able to sequence their own individual human genome. This will be a boon to researchers trying to understand how our individual bodies work and ultimately allow customized medicines and customized anti-aging therapies to be crafted specifically for each of our own individual bodies. Human genome sequencing has already led to breakthroughs. For example, researchers found a specific gene associated with developing age-related macular degeneration in elderly people, which leads to blindness. This gene was previously thought to be unrelated to vision. Clinical trials are also currently underway to fight the AIDS disease by turning back on a patient's T cells, which are normally attacked and shut down by the HIV virus, while also simultaneously disabling the HIV gene. Researchers have been able to successfully accomplish this in cultured human tissue within a laboratory. They are now trying these techniques on human patients. Genome sequencing technology will also lead researchers to discovering weaknesses within cancer cells, making possible the creation of individual personalized treatments. Right now, there's a genome sequencing project underway that studies centenarians, people who live to 100 and beyond, to see if they possess certain protector genes that regular people do not. These superagers, as centenarians are called, appear to have a genetic makeup that allows them to better avoid cardiovascular disease, insulin resistance, high blood pressure, metabolic syndrome, and so on. The key, of course, is to identify exactly which genes keep them so healthy. There are also some amazing experiments currently being done on simple animals that greatly extend their healthy lifespans. At the University of California, San Francisco, teams have partially disabled a gene called the DAF2 gene. This doubled the life of worms. That same lab later altered the DAF16 gene in worms. Those worms lived 6 times longer than normal. That would be the equivalent to a human living for 500 years. Dr. Kenyon, the research team's leader, stated, "Our discoveries have led to the realization that the aging process, like everything else in biology, is not under exclusive regulation. In this case, the regulation can clearly be altered, making aging, once thought to be a process set in stone, malleable." Dr. Kenyon continued, "People have always thought that, like a car, body parts eventually wear out. But we have found that over time, when a single gene was manipulated, a worm actually remained youthful in all ways so that age-related diseases were also postponed." Researchers in Spain have been able to extend, through genetic manipulation, the lives of mice by 45%. That Spanish team accomplished this by turning up the gene that affects the production of an enzyme called telomerase C. This enzyme keeps the ends of chromosomes from shortening. That, in turn, extended the lives of those mice by an average of 45%. As human genome sequencing gets more affordable and faster, this will undoubtedly allow for a much better understanding of our complex biochemical processes and holds the promise to repair problems at the genetic level before they ever occur. All of this is placing us at a major turning point in human history. We are indeed beginning to turn back the aging clock. The goal of stopping aging is the scientific challenge of our time. It's unclear exactly when we will be able to significantly extend human lifespans, but few dispute this will happen. Major anti-aging breakthroughs will occur within most of our lifetimes.